And we're live. Hello, everybody. Welcome back to another edition of Home Lab. Okay. So today we're going to be doing the kidney transplant lab. Okay. This is a rather interesting experiment. Okay. And this is by far the most elaborate we've had so far. Okay. As you can see by the screen next to me, there we go. There's three setups, right? So on your kit, you should get all the material necessary to carry out part part two, and part four of the experiment. These are what we call the wet labs because they include all the solutions, all the pipetting, all the actual procedures. Part three, it's actually just a comparison of the actual patient, the donors, against three different graphs that tell you what is the likelihood of survival for a donor, right, provided their age, weight, and I believe exercise. Okay? So, as always, right, let's be very careful when we're doing any of these wet labs, okay? Specifically, you see my setup right there, right? I have an entire black mat. I divided it into three stages. I do not mix any of my contents. I use styrofoam boards. These are just, the very tops of, the food containers they sell, right? And I just poked holes so that everything can stay in place. Those are not provided, but you can get creative, right? Remember, whatever you use for this experiment, do not wash and reuse, simply toss it, okay? Toss it, all right? I personally do not like using like any Tupperware that I'm going to sacrifice for, like from kitchen or stuff like that. I like to have something disposable, okay? Do not wash this, just toss it out. Okay, next, waste bin, right? There's a lot of little plastic items in here, and we're going to need a waste bin. And for what I have right here is an actual pickle jar, right? You can see it right there. That's my pickle jar, and it's going to be my waste bin for today. All right. So the way I broke this down was on my tabletop, I put everything I need for experiment number one, okay, which includes samples of simulated urine as well as all the testing. I'll go through each one of the contents individually. Then we have sample two or test number two, which is the blood typing lab. This is by far the most relevant with regards to our discussions in lecture at the moment. Okay, because we're dealing with blood typing, we're dealing with blood, cardiovascular system, etc. Okay, the fourth is going to be a white blood cell analysis. Okay, and this one is the one that has the fewest material. So only two test tubes, only two pipettes, and a testing strip. Okay. Notice right here that I kept all the insets, like all the inserts. That's what I call it, all the inserts from the kit because that's what has the actual instructions. So do not discard them. Okay. Do not discard those little instructions because they're very concise and very effective. Okay. Notice also that I kept the little baggie, right, on the test strips because test strips, the material they're actually made for, they have chemicals that if you simply touch them with your bare hands, you may be compromising the results because you leave residues or whatever's on the tabletop, right? It's not sterile, so you want to keep them as sterile as possible. Okay. I also kept one of these bags, right, to show you, right, every single thing in here is not meant to be eaten. Okay. That means that if you have little ones at home where you're performing this, make sure that you're very careful in setting it up and cleaning it up. Okay? We don't want any accidents to happen. All right. That said, okay, please heed all the warnings that are in here. Read them before, right? Essentially, it says, wash if it's still in the hand, if it's going to the eyes, if it's swallowed, go to a physician. Okay, but definitely read it. All right. So here we go. Right? Warnings, right? Read all the instructions carefully. This is a simple lab, right? If you follow those simple instructions, there will be nothing wrong. Okay? Work in a clean area, and like I said, never work in your kitchen or anywhere where you eat. Okay? Garage, a desk, a piece of cardboard, that's awesome. Okay, cover your workspace with paper towels. Okay, anything disposable. I use a top that I can clean the craft table. Okay, do not use the contents of this kit for anything else. Even if you say, whoa, this little vial right here, I can use it for this. No, just toss it. Okay. Goggles or safety glasses should indeed be used. I will be using these as I work on it, right? And as you can see, gloves are a must for this lab. So please make sure that you get some gloves, right, for your hands. I will remind you of this during lecture this Tuesday, but make sure that you get some gloves before you do this. Okay? There are pipettes, which if you're careful, you're not going to get anything on your skin, but I always like to be prepared. All right. Do not drink. Do not eat. Do not have food around whenever you're doing this. Okay? Labs are messy. You don't know what's going to happen. Therefore, it's better to stay safe. Even gum or water, not allowed, guys. Closed-toe shoes, definitely a must as well. Okay? And lastly, when you're cleaning up, make sure you wash your hands, right? Dispose of your gloves, dispose of all the materials carefully. Wash your hands again. Call it a day. All right? If you need to toss your station or wash it up, do so. Okay? All right. Part number one. Okay, materials, right? We're going to need simulated urine samples. Now, this is not real urine. This is actually a solution that has been stained, but it has some, chemicals that will react to whatever we're testing. Okay, so don't be grossed out. We need the urinary test strips, which are designed, right, to essentially determine whether or not there is solutes within our urine. Okay, so we have ketones, blood, protein, and glucose. All right, these are the ones that we're testing for. Okay, and the test is relatively simple, right? All you have to do is dip the strip into each sample of simulated urine, and based on the color, you're going to be able to record and see what's going on there. Okay, I like to keep a plastic underlining on this one, right, just in case, because the strips need to rest a little bit, right? So I'm gonna be using the cap of my pickle jar, right, to actually just set it down as I record my data, and then toss it. Okay. Um, Waste bin, a must. Okay, for all of these. Procedure, like I said earlier, right? You take one of your strips, you dip it in the urine, right? Simulated urine, right? And then you'll see the color change. Compare it to the actual gradient or standard they give you, and record your data. Repeat this for every single sample. An important thing to remember, do not use the same strip for every sample, okay? One per sample, okay? Keep everything separated or everything organized, okay? For part two, this is the blood test, okay? So we're gonna need the patient's blood, simulated blood, of course, the donors, which have one, two, three different people, okay? Antibodies, okay, which are, in short, the molecule that will bind, okay, to whatever antigens are in your blood cell, okay? And if that happens, you're going to cause some clotting or agglutination, okay? A little bit more on the next slide, okay? We're going to need disposable droppers. We have tons of them. Notice that they are labeled, okay? Do not cross-contaminate, okay? Use one for the respective blood sample, okay? Stirrers, you know, because when we're going to add the blood and the antigens, we need to stir it, right? We have multiple stirrers, do not cross-contaminate. As soon as you're done and you record your data, in the waste bin they go. Hence, we need the stirrers and the waste bin. Another important thing is the blood typing sheet. This thing. Okay, you notice that it's plastic right here. Now for this, it's important that you guys put a white paper behind it, white piece of paper, okay? Because if you put it against a black background, it's going to be very difficult to tell any results, okay? Now procedure, okay? So you're going to take the dropper and carefully put about, I say 5 to 10 but a few drops, five should do it, should do the trick on the actual circle. Do not press on this super hard that like everything spills because then you have a huge cross contamination problem. Okay, slowly, right? Extract it, right, with the dropper from the sample vial, and on the actual sheet, one drop, two drops, three, five drops. Keep it nice and neat. Okay. After that, okay, we're gonna add it to two separate wells, right? Make sure that you have enough for both. Okay, and then you add your antibodies. Okay. Okay, patients in their blood. Oh, okay, yeah. So you add blood first, antibodies. I think this instructions are incorrect, but make sure that you read the ones that are in the actual lab manual. Those are absolutely correct. Okay. So the whole idea here, right? And I'll repeat it once I'm actually doing this. Is that we're looking for agglutination. So agglutination is a cloning event. We'll talk a little bit more about this in lecture. But what happens is that antigens and antibodies bind, and you get a grainy type of image. Okay, so on your little plastic paper, you should see dots. Okay, and if there's a print on the white paper, even better because things become so coagulated that you can't even read the words on the other side. It's kind of cool. All right. Now, if you see this, that's an immediate telltale that that is the blood type you're looking for. Okay. So if this happens with anti-A, that means that the blood type is A, right? If this happens with B, that means that the blood type is B. If it happens with both, it means that it's A, B, both of them at the same time. Okay. If it's clear, however, that antigen isn't present. Okay. So the blood type cannot be that one, right? So we're going to go ahead and perform this for every single donor. Okay. Record the results, right? And then see which ones match. Okay. Because remember that recording is one aspect. Then there are a few questions that ask you to conclude something based on the results you observed. Okay. For part three, this is the comparison against the graphs. For that, okay, you're only going to need these graphs right here. So study them, okay, and answer accordingly. For part number four, this one, you're going to need the white blood cells or the simulated white blood cells, right? Two, one for a donor, one for a patient. Two droppers, right? And the actual cardboard or paper strip. Now, this paper strip is what we're going to add our uh, blood blood type directly to. Okay, so what happens is that this paper is not regular paper. Okay, so don't write on it. This one is essentially going to change color, right, depending on which uh, which antigens are present. Okay, or which white blood cells are present. Okay, and once you do, okay, you're gonna seal the pink, and you can go ahead and record it right on this chart. Okay, notice that this and this are identical. Okay, that's a really bad comparison. There you go. So all they're asking you to do is to take the results from here and transfer them onto here. Okay, and that's it. Okay, so that is the procedure. Again, it's very relatively simple. I went over it on the previous one. But as always, you have a little insert for every single lab that gives you the detailed description. So I encourage you to follow that. Okay? As, as they're done, we're going to go ahead and clean up. But for now, let's go ahead and talk about, right, the first experiment. Okay? So we're going to transition now to, all right, so you're seeing right here, this is the stage that I'm going to be using to work on my labs. Let me go ahead and get my safety goggles on. Okay, and let me get situated with this PowerPoint in the right place. And here we go. Procedure number one. All right.
Here we are. Okay. So we're going to go ahead and put this away for now because we are not going to work on that part just yet. Okay. We are going to work, okay, with the simulated urine samples. Okay. Now, notice that you have patient as well as the three good Samaritans that are willing to give this person a kidney. Okay. So there's X, Y, and Z. So these are different people. Okay. And if you read the actual manual, they tell you that they're actually related. Okay. Alrighty. So the first thing we need to do is assess the health of the donor of the donor's kidney. Okay. So to do that, we're going to go ahead and unplug one of these. Okay. Notice how nice this is, right? Just having something to set them up because these have a uh, angle bottom, right? It's very difficult to stand them. As a matter, you can't stand them up. So I recommend that you set them up in something like this, nice and sturdy. Okay. Then you go ahead and take your strip from here, open it up. All right. My hands are nice and dry. Okay. Take one at a time. There we go. Okay, you can let them rest right there. Careful that they don't touch anything else because they're very sensitive. Okay, here we go. This is the test strip. So we're going to dip this part just directly onto the urine right here. We're going to go ahead and repeat that. Okay, and here we go. The instructions say that we should let it be there for 1 second. Now, this is what I mean. This is why you save your inserts, right? You're supposed to compare your results, okay, to that of the inserts, okay? Notice that if you're looking at it under the right orientation, the bottommost shows glucose, second protein, the third blood, and the last one from the top is ketones. Okay, so these are things that you do not want to see in the person's blood or in the person's urine. I'm sorry. Okay, so if the person has them like this, then there's an indicator that their kidneys aren't working correctly. Okay, so we're going to go ahead and leave the patient one here. Notice that there's no mess. Okay, and let's go ahead and cap this one as soon as we're done. No need to keep it open. And we're going to open this one right here. Okay, this is sample X. This is the first person. So let's take another strip. Okay, and we're going to dip it for 1 second. One. Okay, and there you have it. Okay, so looking at this, right, this is definitely within the normal spectrum. Okay, so already we can determine that donor X is looking like it's okay as far as being a donor. Okay, we'll let it settle over here. You go ahead and repeat that for each one of your donors. Okay, I'm not going to do all of them here for you. I'm going to go ahead and expect you guys to complete the rest. Okay, so as soon as that happens, yeah, you keep this recording. Now, if you want, right, I encourage you to keep your strips like this in order. Okay, if you mix them up, there's no way of for you to know which one is which. Okay, therefore, right, be very, very organized. Okay, I'm gonna go ahead and put the rest of these strips back on the little baggie, and I'm gonna show you the procedure for the second experiment. Okay, so we're gonna get rid of this urine samples right here, or I have to keep telling myself it's simulated urine, guys. Okay. Next is blood typing. Okay. Again, we keep all the little inserts because they show you what to do. Okay. Sometimes even better than I can. Now, in here we have all of these, okay? And these are stirrer, guys. So this is what we're going to use to stir our blood types. And you may ask, that's a lot. But in reality, you're probably going to need all of them, okay? Now you bring your little white background over here, and you take your sheet, make sure that you can read it, right? And I'm going to bring my disposable jar over here because I know this one is very messy. It's not messy, it's just it generates a lot of waste. Okay, so here we go. Notice that the first one is patient blood. Okay, so we're going to take the vial with the patient's blood. Okay, open it up, right? No mess. Make sure that if they've been these samples have been tossed around in your little baggie, um, that you let them settle for a bit, okay, before you start working. If there's a lot of blood down here or up here, it may get messy as soon as you open it, so you don't want that. So just let them settle. These right here have been sitting in here for about an hour or so while I was getting everything prepped. Okay, so now that we are going to blood type our patient, let's go ahead and take the correct dropper. Okay, notice that it says patient blood. Okay, so let's go ahead and open it. Okay, remember that we only have one of these, so just squeeze it lightly and extract it. Okay, if there are bubbles, that's fine. Okay, then we're going to go ahead and add a few drops. One, two, three. Three drops should be more than enough. One, two. If you want a third, you can get it. Three, there we go. Any excess, you can just return it. Okay. Now, this guy right here will no longer be used. Therefore, into the waste bin it goes. Close it up, put it back. Okay. Now, we need the antibody. Okay. So each antibody has their specific dropper as well. So we have antibody A and antibody B. Okay. So let's go ahead and start with A really quickly. All right. So we take our A antibody. Once again, we make sure that there's no spillage or droppage over here. Open it carefully. Okay. We take our dropper and we simply extract it. This is a clear liquid, so it may be tricky to see how much you have, so be prudent. All right. So here we go. We add one drop to each. Careful not to make any contact, okay? If you make a contact with it, then we're gonna run into some issues with this because we're gonna need to wash this or something. So make sure that this doesn't touch the blood right here. Now this is gonna go back into its respective place because we're gonna keep using it for every single donor, right? And notice that I actually made a mistake here. I made a mistake and I added antibody A to both A and B. Okay, so how do we rectify this? Okay, if this happens, like it happened to me right now, well, that's why we have a little bit more of our patient's blood. Okay, so I'm gonna fish out my patient's blood. Okay, and I'm gonna extract an extra drop or so, and I'm gonna put it somewhere here. Okay, just one drop will be enough. Okay, this is to rectify my mistake, obviously. And then I'm gonna go ahead and take my antibody B. Okay, and I'm going to add it to the outside area. Okay. hmm Everything else goes back. Okay. And suddenly, if you make this mistake, there is no problem. Now, obviously, whenever you make an error like that, you need to write it down somewhere. Okay. So that you know that this right here corresponds to antibody B, and that this is essentially a replicate of A. Okay. That is if you make that mistake. So notice over here that we're getting coagulation already on the antibody A. Okay, you see how this is not clear, right? It's kind of cloudy, and that's what we call agglutination. Okay, normally you should be able to see clear through the blood sample, but you can't. And we can confirm that we did this right because once again, right, we have this one as a replicate. This is our error. Notice that I can do this right here because these and these are the same solution, right? Is antibody A and antibody A because I made a mistake. Okay. Now I'm going to stop getting rid of that. Okay. Now for the antibody B, remember that I did this to rectify my error. Let's go ahead and stir it a little bit right here. And it looks like to me that this is actually clear. Okay. So if this is clear, yep, it looks clear to me. Okay, if this is clear, then we can go ahead and record the blood type for this patient. Okay, so how do we do that? So we know that agglutination happened, and remember this is our anti-A, and this is our anti-B. Okay, right here. So anti-A definitely has coagulation or agglutination, but anti-B doesn't seem to happen. If you want to do a quick test because you're unsure, you can just run the letters. You see that? You can actually read the letters anti-B through the actual blood sample. But right here, you can't. Right? So definitely B is not present. So we say that B is not here. Okay? That means that the person's blood type is A. Okay? Because only A resulted in agglutination. Okay? So this should tell you right here that our donor should have a blood type that matches our patient. Because if it doesn't, then we run the risk of rejecting our donor. Okay? So that is how we go ahead and complete this stage. Okay? You repeat the process for donor X, for donor Y, and donor Z. All right? One after the other. And remember, okay, pay attention when you are filling this up, right? This one, for instance, right? You can see that all the blood, all the simulated blood solution was like pooled at the bottom. I want to give it a shake, all right? You gotta make sure that it's nice and dissolved in there, okay? Because these are prepared solutions, so always make sure that they're like this and not clearly white for the actual donors. Like look at this one, donor Z. All the good stuff, right? All the solutions are down here, or the solutes. So you have to bring it into solution. Okay, and there we go. Okay, so that's why you should, definitely keep an eye on this. Don't just open them right away. Looks like blood sample Y is doing okay though, like perfectly fine. Okay, so once again, you take one of these, right? If for some reason you stained your glove like I did, be very careful, okay? So no cross contamination. Take your Y, okay? Add it in the right area. 2 drops should suffice, okay? Put this back. In the disposal they go. Put that back. Get antibody A. Get the antibody A pipette, okay? Put everything back that's an excess. Okay, get B. Put all the excess back. Close this up and take a look. Okay, once again, new donor, new stirrers, okay? So let's go ahead and stir it. If you're confident you can stir with both at the same time, just make sure that they don't cross contaminate, okay? As soon as this is done, you can go ahead and all of this go in the bin. And you can carefully glide. If you want to write like test right here or some other word, right? So you can run it underneath. You can see B isn't there. Okay. A isn't there either. So for this person, for donor Y, okay, in terms of A and B, neither are here, which means that this person is O. Okay. That should not happen, guys. Okay. However, this is ultimately what you don't want. Okay. All right, if this happens, what I recommend is for you to completely clean this up using a paper towel, right? A damp paper towel, clean it up, and you can repeat the experiment. They provide a plenty amount of sample blood, so you can actually repeat it. The only limiting factor would be your antibodies. Okay, so again, be very careful. But for us, for the sake of this demonstration, make sure you do X and Z on your own. Okay, so we're going to get this one out of the way. And once this one is over with, right, you can go ahead and carefully grab it. Make sure that it doesn't bounce and splash you. Okay, and into the bin it goes. There we go. Same thing for this trash. Okay, perfect. Clean. The last experiment that we're going to test, okay, pertains the white blood cells. Okay, notice that once again I have my samples here, and I'm going to have my HLA tissue typing screen. Okay, so I'm going to take this is posable donor. Let's start with patient. There we go. So these are the white blood cells of patient. Okay, remember this is simulated. So leave it at that before we open it so that nothing spills. Okay, and the whole idea here, and this is patient, is to add 1 drop of this into each well. Okay. Okay. If you want to put something underneath before you do, in case there's like spillage or anything like that, right? Like a paper towel or a plastic screen, you may. Okay. So let's go ahead and do this. 1 drop. Okay. 1 drop. See that one? You may not be able to see it because of the size, but this color has changed to pink. That's because there's an indicator on the actual paper. 1 drop, 1 drop on each. So all that we want to record is which square turns pink. Okay? Because that is an indicator of the presence of your white blood cells. And the whole idea here is that your donor should match your patient on these as much as possible. Okay? Because if the donor has a lot more of these white blood cells than the patient, then the patient will recognize the donor's tissue as foreign and thus will reject the transplant. Okay? If, however, they match, right? If they have a similar signature, so to speak, it is more likely that the transplant will be successful. Okay? So notice that this, right, kind of dries up quickly, but it definitely gives you that nice pink coloration. Okay, let's bring it up to the camera so that we can appreciate it right here. So we record all the pink and simply transfer that information onto our piece of paper using a pink marker. Okay? Or any other color would work, to be honest. Okay? As soon as this is done, right, and you have recorded everything, into the bin it goes. Okay? If you want, if you're curious, you can even bring these back, right? Because they've been sitting here and notice that they haven't changed over time. It's a pretty good indicator. Okay? But remember all of this also in the bin. Okay? This is my under knee. Okay? We can take them out, right? Be very, very careful, right? And you see, all here. Same goes for this. As soon as you're done and you're satisfied with your experiment, okay, you can go ahead and dispose of each one of these, okay? Because once again, we do not need them anymore, okay? Same thing goes for the simulated urine, okay? If you have these, after the experiment is done, dispose of each one, okay? If you would like to recycle the plastic, okay, the best way to do it is to bring your waste disposal to the lab at Cypress College. Okay, if you speak to one of our lab technicians ahead of time via email, I'm sure they can schedule a pickup so that they can get rid of this and dispose everything, you know, in a more thorough way. But this is totally safe to go on the bin, guys. Okay, no biohazards here. Otherwise, there would be biohazard signage on the package. Okay, and that's that. Okay, perfect. Okay, now let's go ahead and take a look at what we did.
All right. So we have just finished the experiment. Okay. Remember, mistakes can happen easily. You guys saw me doing it, right? I was distracted because I was narrating, right? And I made two mistakes. One was simply keeping track of which antibody I was using. Okay. Don't do that. Be focused. Okay. Second was instead of placing things, right? I simply dropped it, and that just creates chaos. In a normal lab setting, putting things around willy nilly is not that big of a deal because the actual materials we're using, right, they're cups, they're actually more solid. This is for the blood typing, it's just a clean sheet, so it's very easy for things to mix. So please make sure to be careful with that, okay? All right, so that was part two, part three, and part four. Okay? So we will talk about this in lab next Thursday. Okay? As always, there will be a quiz associated to this by the end of the week. So please make sure that after you perform the experiments, you write good notes. If you have any questions, bring them over to lecture. We can go ahead and discuss it. Thursday, this video will go live, right, and the assignment will go live shortly after. Okay? Thank you very much for your time. See you again in class.